This conference will now be recorded.
Okay, so just as a update, talking about what's going on in the course. Of course, Friday is our first exam, so the study guide went up for that last Friday. The way those are going to work is those are going to be online. Again, we're trying to minimize the amount of papers handed out and handed in. Whole pandemic thing and all that. So those will be essay questions posted Friday. They're due Friday. You can do it whenever you want to, okay, but there is an hour, well, 50-minute time limit on it. So you can't start it and stop it. You can't do a little bit and come back later. You need to sit down somewhere where you have 50 consecutive minutes to come in and take the exam. Did my level best to try to keep it to a fairly even balance of muscles and special senses. So there should be roughly equivalent amount of points, if not number of questions. It'll focus mostly on physiology questions. There will be some anatomy on there, but it's mostly physiological processes. The lab is really where we do the anatomy. So bear that in mind. Trying to think what else. It's in Canvas. It's a Canvas quiz, so there's not going to be any other places to go or links to click or whatever. It'll be all in Canvas. You guys have any questions about that? Okay. All right. And like I said, anytime on Friday you want to take it, you can take it. We won't be meeting on Friday. So if you want to take it during that time period, that's great. I would recommend not doing it on your phone if you can avoid it. I understand it's not avoidable for everybody, but it would be easier if you didn't try to do that. Okay. So we're still in the middle of trying to get through muscle and muscle physiology here. One of the things to keep in mind as we talk about contractions, we talk about force, we talk about tension, is that ultimately at the sort of its root level, we can talk about the number of fibers and how big those fibers are, but ultimately what determines the amount of tension being applied is the number of cross bridges being made, being formed. And so you can then extrapolate out from there, right, how recruitment or fiber size or any of those things might play into that. But it always comes down to number of cross bridges. Now, there is one aspect here that we haven't talked about. I can get my cursor. There we go. It's this last one here, this notion of this degree of muscle stretch. We'll talk about that in a little bit. We kind of are going to see kind of a peak of muscle strength that may or may not actually occur where you think it might in terms of the length of that muscle. So to rehash, to revise, to revisit some of these other things, right? As recruitment increases, right? Tension increases, larger individual fibers, more myofibrils, more cross bridges being formed, more force. As we increase the number of action potentials, right? We start having wave summation coming into play. That's what this is getting at. Again, more force. But that stretch one, as it turns out, and really it's at about 100 to 120% resting length. We are strongest when the muscle is at or a little beyond resting length. And as we get beyond that in terms of overstretching it, or as we get shorter than that in terms of over contracting it, we actually lose power fairly significantly quickly. And if you're a visual person, this picture is in your book here as a means to try to explain what's happening there. But again, this last one, I'm going to show you a slightly different picture of that in terms of the stretch. If we look at the bottom of this graph, so this is percent resting length, right? So when your muscle is at resting length, it's right here at 100%, right? And as we stretch it, right, we go this direction, and as we contract it, as we make it shorter, we go this way. If we look at strength then, amount of maximal tension we can apply, which is on the y-axis here, this part right here, the most strength you can bring to bear, most force you can apply, is again in this 100 to 120% resting length zone. And as we go away from that in the other direction, we start to fall off pretty significantly. The reason here is maybe dumber than you might think it is. In order for a myosin head, a cross bridge to form, the myosin head must actually be able to make contact with a thin filament, right? And so as we stretch past about 120%, we actually have a lot of these myosin heads that are reaching out into space. There's nothing for them to grab, right? And so they can't do anything, and there's no force that they can apply, right? So as we stretch past that, we run into that problem. As we go overly short, okay, and this is exaggeration here in terms of the relative position of things, but as we go overly short, we run into kind of the same problem in the opposite direction, where we actually move these thin filaments out of alignment with the myosin heads because they're being kind of displaced by other ones. They get too short. Okay, so again, we have that same problem where we don't have the ability for these guys right here, say, to make contact. And when they can't make contact, strength falls by the wayside. And you can imagine that this is, I guess you can imagine a situation where this would be true, right? If you're doing any kind of lifting or anything like that, you certainly feel strongest at the beginning of that exercise, right? But as say if you're doing a curl, right, as you get to about this point, right, it starts to get real hard, right? And usually if you watch somebody who's lifting too much weight for what they can do, they'll get part way up and they won't be able to go any further, okay? And the reason for that is not that gravity suddenly magically gets stronger, 6 inches farther off the ground. It's that they're simply moving their myosin heads out of alignment with the thin filaments. They can't pull on it anymore, and so they're losing strength just by the shortening of that muscle. So again, sometimes it's counterintuitive, but that's really all that's getting at. So we could talk about force, certainly, and we have talked about force quite a bit, but there are other physical aspects to contraction that are worth visiting. The speed of contraction, how quickly we can actually shorten a muscle, velocity, or how long we can hold that contraction, duration here. And again, those are going to be influenced by a lot of the same things as force, right? So the muscle fiber type in a slightly different way rather than cross-sectional area. It's a slightly different thing that we'll talk about, but same idea applies. The weight itself, right? So what are we trying to move? And recruitment. So that muscle fiber type, what we're getting at here, again, is not size, but how are they built, so to speak. If you remember when we had the twitch myograms up, and we had 3 different muscles, right? We have an extrinsic eye muscle, we had gastrocnemius, and we had soleus. And those 3 muscles, right, all had different periods of contraction. They took longer or shorter amounts of time to fully contract and to fully relax. That is getting at, or that represents here, kind of what we talk when we talk about slow or fast fibers. The faster a fiber is, the shorter that period of contraction is. The quicker we achieve maximal contraction, right? And the slower it is, the longer that period happens to be. We're just kind of stretching out or shortening that curve out on a myogram. And there are, underlying root causes of that. The rate at which they split phosphate groups off of ATP, handful of other things, the rate of electrical signal transduction, stuff that I'm not overly worried about you remembering. But one thing that is worth remembering here is that they have preferred methods of ATP synthesis. So on Friday, we talked about the idea that we can do this either aerobically, which takes time, a fair amount of time, but is very efficient. We churn out a crap load of ATP for every molecule of sugar, right? Or we can do it anaerobically, right? Where we more or less just kind of cut the process off in the middle, and we don't make a ton of ATP, but we do it really, really quickly, okay? These two kind of competing methods here, preferred methods, correspond very quickly or very, very tightly here with our slow versus fast fibers. So if we're going to use the aerobic pathway, the slow one, right, odds are pretty good we're not talking about a fast twitch fiber here, right? On the other hand, if we're talking about glycolytic fibers, okay, where they are using glycolysis, this is going to be a fast means of ATP production, and so is more suited to, say, a fast twitch kind of fiber. And so, right, we might go ahead and start talking about slow oxidative fibers or fast glycolytic fibers. There is one in the middle there that you will no doubt have noticed, the fast oxidative ones. Those guys are special. We'll talk about those a little bit later. Those are not, you're not really born with those so much as you manufacture them through training. And generally speaking, even a fast muscle or a slow muscle has some of all these types. So even if, like, you know, an extrinsic eye muscle will have some slow fibers in it, right? Soleus will have some fast fibers in it, but generally speaking, the majority of the fibers within a muscle will dominate based upon what we ask that muscle to do. Yeah. So I'm not going to walk through this. There's a lot of information on here, and probably it would take us a long time to get through it. But as you're going back through and trying to study for this and kind of parse all this together here, this table is a pretty good place in terms of being a one-stop shop for a lot of that information. In terms of a slow oxidative fiber, you know, what's the rate of contraction? What's its preferred method of ATP production? All that kind of stuff is in there. And there's a lot of extra stuff in here that we're not going to worry about. But so again, don't spend too much time with it, but just as a basic set of ideas. A couple of things that are worth pointing out though, right? So we talked about speed of contraction, primary pathway, right? But there are a couple of other things in here that are worth visiting as well. And that has to do with myoglobin and glycogen. So myoglobin, right, is a protein that binds oxygen. So much like hemoglobin in your blood binds oxygen, myoglobin binds oxygen in your muscles. So we're going to have a lot of myoglobin in muscle fibers that need a lot of oxygen, right? So if we look at our oxidative types, both slow and fast, right, there's a lot of myoglobin there, okay? But a glycolytic fiber, because it doesn't really rely on aerobic processes, doesn't really need oxygen, right? And so we're not going to have a lot of it. It's going to be very, very little. On the flip side, if you're going to be doing glycolysis, your limiting factor is glucose, right? And so there's going to be a lot of glycogen stored in a glycolytic fiber and relatively little if you're in an oxidative fiber, right? So again, you've got to think about trying to pack all this stuff into one cell. If you have a lot of one thing, you have to kind of give up space for the other, right? And so here we kind of pack in the most of whatever we're going to be the most short on, oxygen or glucose. The other thing in here, I guess it's worth talking about, mostly because it's fun, is the color associated with each of these muscle fibers. The slower fibers, the oxidative fibers, are going to be more of a bright red color, and the faster glycolytic ones are going to be more of a pale white color. And this again is the same, it goes back to whether or not there is myoglobin in there or not. Just like hemoglobin, when bound to oxygen, gives you blood a red color. Myoglobin, when bound to oxygen, gives your muscles a red color. So the next time you're at Popeyes and you get a 3-piece meal, right, and you get your drumstick, and it's all dark meat, and you get your breast meat, which is mostly white meat, right? This is why, right? Breast meat is flight muscle in a chicken. Believe it or not, chickens theoretically can fly. And so that's high intensity activity, right? So that's going to be all this glycolytic muscle. Leg meat, on the other hand, chickens walking around, they're not winning any races or anything. They're just walking around. That's going to be more of your slow oxidative type stuff. And so we'll find dark meat on thighs and drumsticks because of what we're asking them to do. Same thing is true in you, by the by. So you do have white meat and dark meat, which is kind of gross to think about, but it is what it is. Okay. So what does this all have to do here with velocity and duration of contraction? Well, if we have a lot of these fast fibers, right, obviously you would expect then that the speed at which a muscle can shorten is relatively fast. And if you have a lot of slow fibers, the opposite would be true, right? I don't think that's breaking any wild misconceptions or anything, right? Same basic rule here is going to apply with terms of duration. How long can you hold that contraction? Okay. If you are built for endurance, right, you have a slow contraction, you have a lot of capacity for aerobic ATP production, you can hold that contraction for a long time, right? So the slow fibers are really good at holding those things. That's why we use them for posture muscles, okay? Your fast glycolytic fibers, on the other hand, because they don't have that long-term efficient ATP production model, can't hold a contraction. So they're fast, but over only short periods of time. Okay, whereas the opposite is true for your oxidative fibers. The last thing that really plays a role in here, right, is this thing in the middle here, the load. Basically, the heavier something is, right, the slower contraction is going to be, and the less amount of time you're going to be able to hold it. I don't think that's, again, too crazy, right? You could probably have worked that one out without me telling you. So if we want to just kind of take a look here at one muscle under several different loads, okay? The heaviest load, we can only contract for a certain amount, or a certain distance, right? And again, we talk about strength, right? As we shorten a muscle, it gets weaker. So for something really, really heavy, we're going to have a limit on how far we can actually shorten that muscle, if we can move it at all, right? And then on top of that, we're not going to be able to hold that contraction for very long. But as we take weight off, right, we can move it farther and hold it for longer periods of time, right? And in terms of the speed at which we can do that, very, very fast, light loads, but increasingly slower as we add weight to it. Recruitment has an interesting role here to play. If we increase recruitment, we can increase the rate of contraction. We can increase the speed of contraction. And the reason behind that is basically we're making a muscle stronger, or you can think of it the other way, we are lowering the ratio of force or load to force, right? We're essentially making it lighter, okay? So you can make it faster the more muscle fibers we bring in. The other role that we've got to play here, okay, is that depending on the proportion of load to maximal force, the lower that ratio is, the longer we can sustain it, because we can turn on, off, we can switch off different muscle motor units. Okay, so the more that we can utilize different motor units to hold the load, the longer that duration is going to be. Now, thankfully, we are not stuck with our genetically predisposed abilities. We have a fairly high capacity for adaptation as a result of exercise or activity. There's a lot of plasticity, I guess is the right term to use there. And depending on the kind of activity that we're asking our muscles to do, they will adapt in different ways. Okay. So if we do long duration, low intensity things, right? We go for a jog, we get on the elliptical bike, whatever. Okay. Not real strenuous in the moment, but for a long period of time. And this is aerobic or endurance kind of exercise. And what we're going to find is that the muscles we ask to do these things, okay, are going to adapt to be better at it, right? Well, what does that mean? Well, if we're going to be doing things aerobically, our biggest problem, right, is blood supply or oxygen supply, really, okay? So we're going to increase the amount of blood supply to a muscle. We're going to make more capillaries in there, so get the volume of blood increased, thus bringing more oxygen in. We're going to increase the myoglobin store, right? So we're going to store more oxygen. We're going to increase the number of mitochondria because, again, mitochondria is where this whole aerobic respiration thing takes place, okay? And in doing so, in doing those things, right, we're going to just kind of take all of the stuff that goes into being a oxidative fiber and just turn it up, okay? Now, we talked before, I said there was going to be that third kind of muscle fiber in there that kind of was in the middle of those two normal settings, right? Something that we would be able to create. This is how we make fast oxidative fibers, okay? If you have a muscle, right, that is fast and glycolytic, and you use it repeatedly in aerobic activities, you can convert it to a fast oxidative fiber. And really, those are the best of both worlds. Our other option here, right, is rather than doing long duration, low activity level exercises, we can do short duration, high intensity activities, right? Resistance training, weightlifting, stuff like that. And because this is high intensity, right, oftentimes we're going to be operating anaerobically, right? So all of the ATP we generate during these activities is going to be glycolytic or creatine phosphate, but primarily glycolytic in nature. And so just like we saw with aerobic exercise, we're going to just specialize on the things that give us anaerobic ATP, okay? So muscle fiber itself, right, we'll start to see increased amounts of glycogen being stored within it, okay? We're going to see increased mitochondria, right? But the big thing, no pun intended, okay, is that we're going to see the muscle fibers themselves get larger, okay? Call this hypertrophy, okay? Hypertrophy above growth. And the way that we're going to do this, right, is we're going to take those satellite cells we talked about way at the beginning of this chapter, and we're just going to start absorbing them. And we're going to start turning them into more myofibrils. And the more myofibrils we build, and the more of them we pack inside of the cell, the thicker it's going to get, right? And the thicker it gets, and the more myofibrils we got, the more cross bridges we can form, and the stronger we're going to be. Now, one other thing I want to point out here, another side effect of doing this is not, nothing in your body is really isolated, right? And so as you do a lot of this activity, you put a lot of stress on the muscle itself, but also on the bones and the connections to the bones. And so while you're doing this, if you're doing this the right way, okay, you'll also build up stronger tendons, and you'll also typically get stronger or thicker bone at that attachment site. If you do it the cheating way and you take a bunch of steroids instead, you'll get muscle hypertrophy without the corresponding increase in connective tissue. And a lot of times what will happen is that will be followed up by a traumatic injury. I cannot count the number of times I've seen people at the gym who have severe vascularity on one side. If you look at Sylvester Stallone, for example, one of his arms is all like fucked up and weird, okay, because he was doing a lot of steroids. And one of the consequences of that is because of the weak connective tissue, he and a lot of people tore one of their biceps tendons, and that then rolls right up your arm like a window shade. Is, I'm told, ungodly painful, but causes a lot of tissue damage inside the arm at that time. So it's something you want to be aware of if you're looking at doing this kind of stuff. Okay. Now, both of these effects only happen or only kind of come to be under what we call the overload principle. And the idea behind the overload principle, it's basically no pain, no gain, right? If you work a muscle, but you don't work a muscle till failure, your body says, oh, that's cool. We've got enough. We don't need to do any more, right? Because muscle is expensive to build and muscle is expensive to maintain. It's only when you prove to your body that the muscle that you've got is insufficient for the task that you actually start to build more. So you really honestly do have to damage that muscle tissue in order to affect change. Otherwise, nothing will happen. Now, with that in mind, then, if you're going to be undergoing an exercise program with the understanding that it's going to involve tissue damage, you need to build in rest days, right? It can't be arm day every day, right? You got to give it a couple days to kind of relax. A good way to do that, right, is to do resistance training some days, aerobic training the other days, okay, and space those things out so that you give your body time to recover, you give your body time to build, because again, the whole point is that you're tearing this stuff down, right, so that you can rebuild it back up. On the other side of that coin, okay, if you do not use muscle because it is so expensive to maintain, you will lose it. The sort of extreme end of that gets into what we call disuse atrophy. And disuse atrophy isn't something you're going to get because you decide that this summer I'm just going to sit on my ass and watch Tiger King and do nothing else, right? This is what happens when you get in a car accident and you have a spinal, right? And you physically can't move your legs anymore, right? At that point, if you are fully and completely immobilized, you will really quickly start to lose muscle, and you'll lose it pretty fast, right? 5% by volume per day is a pretty rough number on that, but it is about what you'd expect to find. And the big thing here, right, is without that neural stimulation, okay? So we talked about muscle tone before, okay? This constant sort of cycling through all the switches in your muscles to keep them all a little bit active, all a little bit ready to go. When you have nerve damage or you have a spinal, right, you're a quad or a para, you lose that altogether. That goes away, okay? And so in the absence of that, your muscles get no stimulation, and you really will quickly lose a lot of that down to maybe a quarter of their initial size, okay? At that point, there is no rehab. Those muscles are too far gone. So one of the things that you'll see a lot is even people that are paras or quads, right, will get stim therapy. They will come in and they'll get some jolts, okay, to try to keep that muscle in case they're able to reverse whatever the damage to the spinal cord is, okay, because they don't want to get down to that point of no return. Okay, so that's skeletal muscle in a nutshell, a 3-day nutshell, but a nutshell nonetheless. The only one other kind of muscle here we're going to spend a lot of time today talking about is smooth muscle. We'll cover cardiac muscle when we talk about the heart. But smooth muscle we might as well get into here because it does things really quite differently than skeletal muscle does, and in some pretty profound ways that are worth having the kind of comparison that we might be able to make. So for smooth muscle, rather than being associated with the movement of the body. Smooth muscle is associated with moving things through the body. So hollow organs, and we're expecting to find this stuff. Basically every hollow organ except for your heart. So blood vessels, GI tract, urinary tract, urogenital tracts for that matter. Basically all of those internal spaces we might expect to find things moving in and out. Respiratory ducts, all that. Typically, we're going to find them in opposing layers. Okay. Sometimes we will find 3 layers, but typically we find 2. And we're going to refer to these as the longitudinal and the circular layer. And the reason that we call them that, right, is because the line of action of each of these layers is set up basically 90 degrees to one another. Okay. So the longitudinal muscles, right, if you have a tube, you might imagine a tube, okay, it's going to shorten that tube, all right? Whereas the circular layer, when it contracts, is going to make the diameter of that tube smaller. So we can shorten the whole tube, or we can make the tube narrower. And we're going to do that again and again and again, and that's how we're going to move stuff through your body, at least in the digestive tract. So if we look at those 2 layers, okay, the inner layer is that circular layer, right? And again, that's going to be the layer that makes the inside diameter, what we'll call the lumen is the technical term for that. We'll make the lumen smaller. The outer layer is the longitudinal layer that makes the whole pipe shorter. With smooth muscle, rather than having one fiber that runs the entire length of the muscle, we have a whole bunch of little muscle fibers, or muscle cells. Okay. And if you think back, way back to ANP1, right about this time, actually, a week earlier than a year ago, but right around this time, we looked at smooth muscle under the microscope, right? And it had a very, I think I still have a picture of it, a very distinct appearance, right? It had these sort of spindle-shaped or spool-shaped cells, right? If you looked at the whole thing, it had this very much this sort of sheet-like appearance to it. Okay, and that is very much what smooth muscle ends up being. It's short, small cells that form big, broad sheets. Okay, they don't have the same degree of packing like we see in skeletal muscles. We're not going to make fascicles. Okay, we're not going to surround the whole thing. We're only going to have an endomysium. So no perimysium, no epimysium, just the endomysium, just the cell membrane itself there. We are not going to rely on the sarcoplasmic reticulum for calcium. We are still going to use calcium, but we're not going to get it from the SR. The SR, when present, is pretty insignificant. Sometimes it's missing altogether. Where we're going to get the calcium instead is from the extracellular fluid. Okay, so the calcium, rather than being inside the cell at all times, is spending most of the time outside the cell, okay, and gets gathered in little pockets, little infoldings of the cell membrane called caveolae, which is what this is right there. Okay, and when we need to bring calcium in for our excitation contraction coupling, right, we're going to get it by bringing it in through those caveolae. There's no sarcomere, there's no T-tubule, there's no myofibril, there's none of those parts, right, which is why they're not striated. Right? Remember, those striations are built because of the arrangement of the sarcomere. None of that's here, so there's no striations. Excuse me. Again, right, these 2 layers that we've got performing opposite functions, shortening the tube, constricting the tube. Okay, again, here's that term lumen, right? The hollow space inside any of our hollow organs is called a lumen. And together, right, by alternating these behaviors, right, by contracting, by constricting, shortening, constricting, shortening, constricting, shortening, we're going to achieve what we call peristalsis, okay, which is sort of the way in which we squeeze things through some of these hollow tubes. It's like squeezing toothpaste out of a tube, right? This is the basic way we're going to do that. And it takes time, right? But it's this rhythmic alternation of those contractions that allows it to happen. Okay. And here's that diagram from before. Again, in terms of this first exam here, really we're going to focus most heavily on everything but this guy right here. We'll come back and talk about cardiac muscle later on. And again, kind of continuing from there a little more detail on that chart. We don't have really any second or third order structure. We just have cells in a big sheet. Nothing like what we see in skeletal muscle. Another big important thing here for us is if you think back again to skeletal muscles, right? One of the big things we had there was that every single muscle fiber had its own axon terminal, right? Never more than one, never less than one, always exactly one. And they were all wired up directly. They had a synapse. Everything was normal in terms of how we understand nervous system connections to be. When we get into smooth muscle, you throw that right out the window, okay? We're not going to have any, not really have any axon terminals. We're not going to have any normal synapses. We're not going to have any one-to-one connections. Most of the cells in a smooth muscle layer are not going to be connected to the nervous system at all, okay? We have something instead that's really built around trying to get, rather than one signal very quickly, around trying to get all of the cells in a sheet to act in unison, okay? And so here our message isn't about getting it to one muscle fiber or one muscle cell. It's about trying to communicate to everybody at the same time, right? And so that's what this is all about right here, okay? We have, instead of a neuromuscular junction, we have what are called diffuse junctions, okay? And these diffuse junctions, rather than being like a normal synapse, are just places where we kind of release neurotransmitter out into the world, okay? It's kind of like, and this is, I realize this is kind of a weird analogy, but it's really the only one I've ever been able to kind of put together. If you've ever seen fish spawning before, okay, they just kind of, they don't have any interlocking parts, right? So when fish want to have children, they just kind of swim around to the same area and just kind of release a cloud of sperm and eggs and hope for the best, okay? This is what a diffuse junction is. There's no one-to-one, there's no meeting of any parts here. We just release neurotransmitter out and hope for the best, okay? And that will contact some of these cells and it will miss most of them. But as long as we get the right ones, as long as we get a couple of them, we're going to be okay, right? Because the other part of this that becomes very important for us, I'll come back to this in a second. If I've got this in here or not. The other part that's important for us, okay, is that in a region, all of these smooth muscle cells are connected to each other by gap junctions, okay? And if you remember from, again, around this time in A&P1, we talked about gap junctions as being ways to essentially make two adjacent cells one big cell. The cytoplasm is continuous between them, so anything that happens inside one cell happens essentially instantaneously in the next one. And so if we wire them all up, okay, in sequence with gap junctions, we only need one of those cells to actually encounter neurotransmitter for them all to become stimulated, all right? And so if we have them all kind of in an area, one big sheet of them all wired together, we can have something like this, and it will work wonderfully. And they will all contract at the same time, okay? Which is how we get things like peristalsis to work. So take a closer look at what this is. Here we have our nerve fiber coming in, right? And rather than having axon terminals, we have these swellings called varicosities. And these are just bulbs, basically, or swellings out full of neurotransmitter, okay? If you're familiar with the term varicose veins, okay, the term there is essentially the same idea. There, varicose veins, there's swelling because of the buildup of blood above a valve, and so the blood vessel stretches. Okay, here we've got these sort of stretching things because we're full of neurotransmitter. Now, once we've released the neurotransmitter, we're going to get a signal inside these cells for contraction. The actual method of contraction is, on one hand, quite similar. On the other hand, really quite different. We're still going to rely on sliding filaments. We're still going to rely on actin and myosin, and all that stuff is still there. Okay, but they're not arranged in one continuous band. They're arranged kind of crisscross throughout the whole thing. We'll see a cartoon of what that looks like in a little bit. And the other part that's different here is the trigger is really quite different. So instead of relying on tropomyosin, troponin for the binding of calcium and the movement of exposure of active sites and all that kind of stuff, none of that's present here. Instead, our coupler, if you will, okay, to bind to calcium is a protein called calmodulin instead. Okay, and calmodulin is going to play the big role there in turning that contraction on. Again, right, the idea we don't have sarcomeres, we don't have myofilaments, we don't have that single tubular arrangement. Instead, we have them spirally oriented, maybe is one way to put it. So crisscross is another way. And they anchor not at one end of the muscle cell and at the other, but all around the muscle cell at little plaques called dense bodies. And those dense bodies are what actually get kind of pulled together when the muscle contracts. So, a little confusing maybe to think about this in terms of text, right? So if we look at a smooth muscle cell relaxed, okay, here we've got our little caveolae, right? So here's where all of our calcium is being stored out here, right? And our gap junctions connecting us to our adjacent cells, our neighbors, okay? Here and there and there and there and there and there, right? We have these plaques, these thickenings of the cell membrane, okay, called dense bodies that are going to be the anchors, right, that these thin filaments and thick filaments pull on as they slide past each other. Kind of looks like a ham, like a Christmas ham, right? It's all trussed up, right? And we've got these little peppercorns embedded in it. It's the same basic idea. And when this thing contracts, right, because it's not pulling at the one end and the other and pulling in one direction, but kind of pulling and twisting all the way around, the contraction of a smooth muscle actually kind of corkscrews it up. And so we get this sort of shortening and twisting this way. It's not a linear thing. It's more of kind of a twisting. So it's a little bit different, right? A little bit different in terms of its actual mechanics. Because we lack that kind of uniformity of direction, right? Because the fibers are all kind of scattered around, because we rely heavily on gap junctions for signal transduction, all this stuff, the end result of a smooth muscle contraction tends to be pretty slow, but coordinated, right? It's like when you're learning to play the drums, right? You can't all of a sudden go at it with blast beats and being crazy, right? You got to kind of start doing things really slow to coordinate it out. Your body never really moves past that stage. So if we want to be coordinated, we need to move things as relatively slow. Okay. The other part about this that's going to be a little bit different, and something that we share with cardiac muscle, is that smooth muscle doesn't even necessarily have to have any connection to the nervous system at all. Right? In fact, there are parts of your body that have smooth muscle that are perfectly happy to be left the hell alone. And the way that they're going to contract is they're going to contract at a predetermined interval. They have parts of their certain populations of their cells that are self-depolarizing. Okay, we call them pacemaker cells sometimes, and they will just, like clockwork, every so often, right? Whatever 15 microseconds or an hour or whatever the sort of time duration there is. Just contract on pace all day, every day for as long as you're alive. Some of them will also respond to things like stretch. So particularly in your digestive system, right? As we move food through it, the organs themselves will stretch, and they'll respond to that stretch by contracting. So we don't necessarily have to have any real connection to that nervous system at all. Okay? All right. That's pretty good for today. We'll finish up muscles on Wednesday, and of course have that exam on Friday. If you guys have any questions about that, please don't hesitate to ask. I will be more than happy to explain it as best I can. This is the first time we've done an exam like this, so hopefully everything will go smoothly.